Good morning, afternoon, and evening, and a warm welcome to our third webinar in this exciting Extracellular Matrix Symposium Series. It's truly a pleasure to have you all here today, joining us from various corners of the world. I hope you are as eager as we are to dive into the world of fibro-inflammation and IBD, where we'll discuss neutrophil activity, mucosal damage, and serum calprotectin. So before we begin, I just want to take a moment to acknowledge the success of the previous webinars on fibroblast activity in the context of heart failure and a webinar on tumor fibrosis. So we're thrilled by the overwhelming response and engagement we received, and we're really grateful for your support. So your enthusiasm and participation and ideas have encouraged us to keep bringing you valuable insights and discussions on the relevance of extracellular matrix in chronic diseases. So today we have an exceptional lineup of expert speakers who will enlighten us with their knowledge and expertise on fibro-inflammation. And so as Morten is unfortunately unavailable today, we'll firstly have the privilege of hearing from Christine Bay-Jensen, Chief Biotechnology Officer of Nordic Bioscience, who over the past 20 years has published numerous articles on extracellular matrix biomarkers and tissue destruction diseases. So with years of experience and invaluable insights, and will shed light on the relevance of the extracellular matrix in chronic diseases and provide a short introduction to the importance of measuring neutrophil activity. And joining us as our second speaker is highly respected key opinion leader in the field of IBD, Dr. Florian Rieder. He's Vice Chair, Co-Director of IBD Section, Director of IBD Research, and Director of the Advanced IBD Fellowship Program at the Department of Gastroenterology, Hepatology, and Nutrition at the Cleveland Clinic. His clinical focus is patients with IBD, with a special emphasis on the field of pathogenesis. So prediction and as well as therapy of intestinal fibrosis. Dr. Rieder has published more than 150 articles, has an H index of 52, and has been recognized for his expertise as indicated through several invitations for clinical guideline steering committees of ECHO. And then last but not least, we are honored to have Nordic Bioscience esteemed head of gastrointestinal diseases, Dr. Joachim Mortensen, with us today. Joachim has been at the forefront of groundbreaking biomarker initiatives to quantify fibro-inflammation, which covers neutrophil activity, mucosal damage, and intestinal fibrosis. These are widely explored in preclinical and clinical studies. Joachim possesses an incredible depth of knowledge of IBD and fibrosis that will undoubtedly enrich our discussion. So throughout the webinar, we encourage you to actively engage with us. So please feel free to use the private chat on the right side to ask your questions or share your thoughts. We'll be selecting some of these questions and addressing them during the dedicated Q&A session at the end of the webinar. So don't hesitate to participate and make the most out of this interactive session. So without further ado, I hand this virtual stage over to our first speaker, and Christine will kickstart our discussion. Enjoy the webinar.
So thank you very much, Major, for those kind words to all of us. And I'm sorry that you have to make do with me instead of Morten. Some would say that maybe this is a better choice, but I will not debate that. Anyhow, I first want to remind that we have that Nordic Bioscience is one of the main sponsors of the Extracellular Matrix Pharmacology Congress that's going to be held in a year's time in Copenhagen, and we hope that you will also look into this. We at least would like that a lot of ECM interested folks attend and follow this society. So I will give a small introduction to extracellular destruction, extracellular matrix destruction, and IBD. It's not my main field of research because I'm more founded in rheumatology and immunology, but I find it interesting how much matrix matters in IBD, and I will talk a bit into that as an introduction to our two really experts in the field. So fibrosis and destruction of tissue and destruction of extracellular matrix remodeling is essential for many pathologies, also for immunology. And I think if we go 10, 20 years back, especially immunology diseases were not thought of as diseases of tissue. They were thought of as tissue, and thought of as diseases of pain, thought of diseases of inflammation. But ultimately, what we find and other find is that all diseases somehow are associated with the tissue. And in the tissue, there are many different proteins. In fact, there are 28 collagens, 46 collagen change. And all of these proteins, including proteoglycans as well, are responsible for tissue integrity. It also provides a ground substance for biomarkers, and it also provides targets for treatment. So extracellular matrix is extremely important, and we have shown that at the previous seminars, and will continue also trying to promote that extracellular matrix should be really the way to target the disease in the future. So basically, what happens in our body on a daily basis is that it's broken down, it's demolished, and then it's repaired, it's maintained, and it happens all 24 hours, 7 days a week. What happens in many diseases is that either the breakdown is halted or blocked, or the formation of tissue is accelerated. So in one case, we have tissue destruction, and in the other case, we have fibrosis. And in some cases, like for example, systemic sclerosis, we also see that the general turnover is actually accelerated. And the question is whether that's also true for IBD. What we see in IBD is that it's not just a fibrotic disease, it's also a disease where we have high level of destruction of tissue. So we could characterize this as a high turnover disease. Other diseases, as an example, to put up the contrast, would be decorative diseases or destruction diseases like RA, where there is a destruction of the tissue that leads to crooked hands, for example, or an OA where loss of tissue leads, osteoarthritis leads to massive pain, or in NASH where you have a fibrotic disease where you build up more tissue that becomes stiff and are not maintained properly. So in IBD, what we have is that we have a chronic flare or chronic inflammation of the intestinal tissue, and that involves the remodeling of the tissue, especially type 4 collagen, type 3 collagen. And what is often seen in here, and what is often which is often difficult to measure by MRI or MRE or endoscopic scoring, is that a tissue can look healthy in IBD, but in real life, what we see that in the deeper layer of the intestinal tissue, we actually see a high level of fibrosis. So this is depicted here, for example, that we have a massive layer of blue color, which indicate that we have a thickening of the tissue, things that we would not see by these kind of techniques necessarily. So it's important to say that even disease that we don't characterize as fibrotic, we actually do see a fibrotic component. So what happens? How does this tissue become? How is it destroyed? So macrophages, T cell, neutrophils, they degrade the ECM. Well, they don't degrade it themselves. They express proteases that's released into the tissue and that fragmentize the tissue, and the tissue is replaced by new tissue, sometimes the wrong tissue in some pathologies. But basically, macrophages, T cell, neutrophils, amongst other, degrade the tissue. They express proteases that does that the tissue becomes destructive and malfunctioning. So today we'll have a bit of focus on human neutrophil elastase degraded calprotectin, so the alarmin is 100A9. And this is an interesting molecule that Joachim will talk much more into, but I will give you an introduction why this is important. So what happens, and neutrophil plays a great role in IBD. It's believed to be one of the main actors in the immune responses in IBD. And what happens is that when neutrophils enters the tissue and it's meeting its pathogen, it forms these nets. And a part of the nets is that calprotectin is released, but also the human neutrophil elastase. And when they come together, they actually generate a small fragment that's then released into the circulation. And that fragment is actually a picture of activated neutrophil, not of the count of neutrophils, but activated neutrophils. So, for example, what we did in one experiment, in vitro experiment, was to compare inactive neutrophils in culture with activated neutrophils, and we measured the CPA9 fragment and saw that this was 100-fold higher. The release of this fragment was 100-fold higher in activated neutrophils. But when we measure calprotectin alone, so the S100A9 subunit of calprotectin, we didn't see that picture, meaning that to monitor the active form of neutrophils, we really need to look at the degradation or the product of the activity. It is where CPA9-HNE is one of those products. So Joachim will talk a bit about that in IBD, but is neutrophil activity only interesting in IBD? Obviously not. I mean, there are other diseases where we see elevated levels of neutrophil activity. We have shown data on COVID that we have extremely high levels of neutrophil activity and release of this CPA9 in COVID. And here I give you some example of IPF and COPD, two lung diseases. And what we see here on the first graph is that IPF patients and COPD patients have significant elevated levels compared to healthy control of this CPA9. So the active a reflection of the active neutrophils. But when we looking at just calprotectin measured in this case by the Buhlmann assay, we saw that there was hardly any difference between disease and healthy. Again, underlying the effect that is important that we measure activity rather than the passive release the neutrophils provides. And we also looked in HS here and looked at how that would look at disease. And what we can see, the blue line on the top here is actually neutrophil activity measured by CPA9-HNE is the one that separates healthy from disease of these severe hidradenitis suppurativa patients to a similar level as we saw with CRP. But of course, these two are of completely different pathways, but also better than the neutrophil count, I should say. So maybe it's complementary to actually counting the neutrophils and then having the activity next to neutrophil counts. So that was the very short introduction for me, just to highlight the importance of understanding the immune cell interaction with the tissues that's affected by the disease. And in this case, I wanted to highlight the neutrophil activity marker CPA9-HNE. And how that's not only in IBD is important, but it's also important for other diseases where neutrophils play an active role, an important role. So, but I'm not the expert, as I said, so I wanted to keep this short to give more time to the experts. And of course, Florian Rieder, I think, is ready to present his presentation on mucosal damage and interstitial fibrosis and IBD, but also Joachim with the fibro-inflammation in IBD. I'm really looking forward to this. And again, I just want to remind that we are setting up several symposium on different topics of the ECM and how that works with biomarker pharmacology and disease pathogenesis. Thank you very much, Major.
Thank you, Anna. Great. Then let's move on to the next speaker, Florian. Anna, if you could stop your presentation and then, oh wait, I can do that for you. Yeah, and then Florian, if you could start your presentation.
Very good, no problem.
Perfect.
Just checking if I can advance. Perfect. Oh, there it was. And it just went away. I'm gonna try again. Here we go. So thank you very much for having me, and I'm very happy about the ECM community engaging in this space, which I think is very important for chronic diseases, and in particular for chronic inflammatory bowel diseases. And I'm going to talk about mucosal damage with a focus on intestinal fibrosis and what could be mechanisms driving that in inflammatory bowel diseases. They do work a lot with TREE to develop a pathway to test anti-stricture therapies in Crohn's disease. We are leading a global effort called the STAR Consortium to do that in collaboration with our industry partners. And this is the topic of today's presentation. And Anne already gave you the background of stricturing disease being a significant clinical problem. This is an ileocolonic resection. You see the ileum on the right, the colon to the top, and the stricture in the middle, the most common site for stricturing disease, which is the terminal ileum. And I will talk about intestinal remodeling being relevant for Crohn's disease and for ulcerative colitis. It's dynamic and multifactorial. I will highlight a few mechanisms such as the microbiota and then inflammation-independent mechanisms in creeping fat. And at the end, I'm going to talk about novel clinical trial endpoints that may allow testing of anti-stricture therapies. So tissue remodeling is relevant for Crohn's disease and for ulcerative colitis. And in Crohn's disease, this case is easy to be made, where you have a relapsing remitting inflammatory disease course that you see here in red. And unfortunately, the slide is not animated in that template. But underneath the threshold for clinical obstructive symptoms, which is a dotted line, you have an increasing fibrosis burden that Anne showed you nice histology from, becomes clinically apparent in more than half of patients with Crohn's disease. The morphologic correlate is seen on the right, top right endoscopy, luminal narrowing, impossible or difficult to pass with an adult colonoscope. And on the bottom right, CT enterography, where you find luminal narrowing, wall thickness, and pre-stenotic dilation as the hallmark of stricturing disease on imaging. The majority of the patients get diagnosed through cross-sectional imaging. Fibrosis is also relevant for ulcerative colitis, and this is an emerging topic. You have in long-standing ulcerative colitis the so-called lead pipe colon, which is stiffened foreshortened colon pipes. When you perform colonoscopy, it's very easy in these patients because the scope doesn't loop due to all the wall stiffness. And interestingly, patients that have long-standing ulcerative colitis, even when you have effective therapies driving complete mucosal healing, the patients may still have significant symptoms of, for instance, urgency. And we believe this may be driven not by changes in wall compliance due to fibrosis. And Alyssa Gordon in our team performed this systematically in 1 year of colectomies at the Cleveland Clinic. These are about 100 colectomies a year. Every 10 centimeters, we got H&E and trichrome stain, and we found fibrosis in 100% of the colectomies with a gradient from proximal to distal. And this is true for the adult population. We recently published the same for the pediatric ulcerative colitis population. In both ulcerative colitis and Crohn's disease, inflammation and fibrosis are intertwined with each other. And there's almost no instances where you find fibrosis without inflammation or inflammation without fibrosis. In both strong reports of areas of fibrosis outside of areas in Crohn's disease or ulcerative colitis that have never been affected by inflammation. But it seems that inflammation is important for initiation of the disease, because if you have fibrosis established and you suppress inflammation, observational data and animal data suggests that fibrosis can progress in the absence of any inflammatory activity. And there's hope because fibrosis of the gut is reversible. It was believed to be a one-way street, but our colorectal surgeons teach us that when you perform a strictureplasty, which you see here on the bottom left, middle, and bottom right are the three most commonly used technique: Heineke, Mikulicz, Michelasi, and Finney. We create a surgical pouch around the narrowing of the bowel. The 5-year surgical recurrence rate is 28%, but only in 3% of the cases. And the recurrence and the reason for the surgery is the site of the prior stricture, because the stricture regresses, and the surgeons, when they go in for other reasons into the abdominal cavity, palpate the area of the prior strictureplasty and identify wall thickness regresses and stiffness regresses. We find it also in serial imaging studies, for instance, ultrasound studies, after strictureplasties. And what this drove in our group and in our global group and local group is we built sketched out a pathway and what would it take to develop anti-stricture therapies, because there are no selective anti-stricture therapies in this space available. And this includes preclinical development with discovery, target expression, in vitro vivo validation studies, but then also clinical trial endpoints are critical with creating stricture definitions, developing the endpoints, validating the endpoints, and then collaborating with industry to get to phase 2 proof of concept trials in this space. But first, let's talk about the pathogenesis, because pathogenesis of wound healing and fibrosis in the gut is dynamic and multifactorial. And the major cell type, just as for all fibrotic diseases, is the myofibroblast that you see in the center of the slide. Myofibroblast is exposed to cell-cell interactions with immune and non-immune cells, such as endothelial cells, epithelial cells, and to their soluble mediators. But it's also exposed to environmental factors, because these fibroblasts are in very close proximity to the intestinal lumen, which contains microbiota and other environmental factors. And how close the myofibroblast in the gut is to the lumen is depicted here. I brought you these stainings. In green, you see E-cadherin for epithelial cells, and in red, alpha smooth muscle lectin for fibroblasts, for myofibroblasts and smooth muscle cells. And they're directly underneath only one layer of epithelial cells. And in ulcerative colitis and Crohn's disease, their number increase, and you have ulcerations and increased intestinal permeability. So these cells are exposed to luminal microbiota. They're not only exposed to it, machinery to sense the microbiota, not 1 nor 2 TLR1 to TLR9. These are primary human intestinal myofibroblasts. We then used highly selective ligands for all of these receptors, and they have a lot of different functions, but interestingly, only flagellin expressed on flagellated bacteria drove a profibrogenic response in primary human intestinal myofibroblasts. When you then delete bacterial sensing selectively on fibroblasts in the gut by inhibiting MYD88, which is an adapter molecule for a variety of toll-like receptors, you can prevent intestinal fibrosis from occurring. So it seems to be that not only epithelial cells and immune cells sense the microbial environment, but in fact, fibroblasts have the ability to do so as well. But what I'm most in scientifically, and I think it has the highest potential for future therapy, are so-called inflammation-independent mechanisms of intestinal fibrosis. And I introduced to you earlier, when you have a flare in Crohn's disease depicted here in red and blue, and you find that inflammatory activity, your inflammatory markers go up in endoscopy, you see ulcerations, erosions. If you treat this patient, inflammatory activity goes down, but the level of profibrotic factors remains high. So there's a disconnect between suppression of inflammation and progression of fibrosis, suggesting inflammation-independent mechanisms. And this is clinical observational data suggests that. And we have a lot of information from other fibrotic diseases outside of the gut, also suggesting inflammation-independent mechanisms in Crohn's disease. Well, this drove in our group, and that's perhaps the reason why I was invited here with the extracellular matrix interest, is we are exploring in a variety of work streams how could the extraintestinal matrix, how could the extracellular matrix influence cell function in intestinal fibrosis. And I give you a few examples, starting with matrix composition. And interestingly, despite strictures being known for about 100 years, so they're part of the first description of the disease, early last century, matrix is present in stricturing disease compared to control tissues. And for that purpose, we developed a decellularization protocol with primary human intestinal resection tissues from Crohn's disease, ulcerative colitis, and controls. And within Crohn's disease, we used strictured and non-strictured segments of the same patient, tested several different protocols, and we pre-specified that we want to have maintained structure and maintained matrix decomposition, all cell and cell components removed, and these matrices we generate have to be biologically active. Then among all the protocols we tested, we chose the optimal one. And when you then take primary human intestinal fibroblasts, which you see on the bottom left, cellularized extracellular matrix, they like sitting there, they attach to the extracellular matrix and in fact align with the extracellular matrix. So the ECM components that we decellularized seem to be biologically active. We then performed proteomics of the decellularized tissue segments, and interestingly, qualitatively, meaning the type of extracellular matrix, there was almost complete overlap between the different tissue pieces, so non-stricturing Crohn's, stricturing Crohn's, ulcerative colitis, healthy tissues. But quantitatively, there were quite some differences. So meaning there's no new matrix molecule that all of a sudden appears in stricturing disease, but the amount of matrix of candidates, we then were interested in exploring MFG8, was upregulated in stricturing disease. We confirmed that on a gene expression level in immunohistochemistry. And the reason why we got interested in it is other than the fact that it's upregulated, because a lot of matrix molecules are upregulated in stricturing disease, but the function of MFG8 on primary human intestinal fibroblasts was in fact anti-fibrotic. So it's an anti-fibrotic matrix molecule that is upregulated in stricturing disease. And you may wonder, so why doesn't it act in an anti-fibrotic manner then? And the reason is that on the right, in healthy fibroblasts, when you perform next generation sequencing in the MFG8, anti-activated, stricture fibroblasts remained largely silent to the actions of MFG8. And you can then subtract the gene pathways from one another, which suggested integrin signaling, then characterized integrin expression, primary human intestinal fibroblasts, and identified with loss of function, gain of function experiments that a specific integrin mediates the action of MFG8. This integrin is downregulated in stricture fibroblasts in situ in stricturing disease. And then MFG8 was also anti-fibrotic in animal models of intestinal fibrosis. The second mechanism I will briefly touch upon is the matrix stiffness, and this is old news to ECM experts like many of but the extracellular matrix stiffness has a profound cell function. The pathways mediating that are known for a long time, but there's a challenge in targeting them. And the best example is the Rho-ROCK pathway that is central for mechanosensing, but targeting it has cardiovascular side effects. To get around this, the gut has unique opportunities because you can locally deliver antifibrotics to the area of the most common site for stricturing disease, which is the terminal ileum. 75 to 80% of the cases, strictures are in the terminal ileum. And then if you generate locally degradable drug formulations, they don't reach the systemic circulation, but have high local concentrations. And that very nice work in an experimental animal model, local delivery of a so-called soft rock inhibitor had antifibrotic properties. And this is a program now and moves into a clinic for drug development. One tissue compartment that gets largely neglected when thinking about stricturing disease is our fat tissue. You may wonder, how does fat tissue in fact relate to fibrosis? In Crohn's disease, you find the phenomenon of creeping fat, where the mesenteric fat wraps around the gut. You cannot find that diverticulitis and ulcerative colitis. You cannot find it in the healthy gut. And is relevant because in about 85% or so of cases, when you have creeping fat, you have an underlying stricture. And so the way our surgeons, when they perform laparotomies, find strictures is by palpating at the areas with creeping fat, because this is where most of the strictures are. So it's a strong correlation between creeping fat and stricturing disease. On the left, you see a cross-section through a human gut. CF is creeping fat, and MP is the muscularis propria, which is the outer layer of the intestine. And we know from histopathologic studies that the thickening of the muscularis propria is the strongest contributor to luminal narrowing. And the luminal narrowing is what drives the symptoms. So it's not the matrix deposition that for the most part drives the luminal narrowing. It's actually a muscle thickening. And creeping fat creeps underneath the serosa directly on top of the muscularis propria. So the two intimately interact with each other, which makes it likely that there is a connection between creeping fat compartment and the muscle. And the increased proliferation when it underlies creeping fat in the gut. And we use relatively simple systems to explore the interactions. We take primary human creeping fat, mesenteric fat from Crohn's disease, ulcerative colitis, and health, condition undernatants, and then put it on primary human intestinal smooth muscle cells isolated from the muscularis propria. And the observation was that creeping fat conditioned medium drove a dramatic upregulation of muscle cell proliferation. But Crohn's, you see, a normal mesenteric fat did not. And this led to an exploration of what factors released by creeping fat could drive this, and we performed lipidomics analyses where multiple free fatty acids are upregulated in creeping fat, with palmitic acid being the one strongest upregulated among those free fatty acids. We identified the pathway muscle cell proliferation, and we just at DDW presented a new fat deletion model where when you take away all fat in an animal model, there's less muscle thickening of the muscularis propria upon chronic inflammation of the gut. But the next question that frequently comes up is, why does creeping fat in fact form? And this is a widely debated topic. And the way we try to get closer to it is, again, by looking first at the histopathology of the disease. This is depicted on the right. You see CF is the leading edge of the creeping fat. MF is the mesenteric fat. When you zoom in to the leading edge of the creeping fat, which is depicted on the very right, and look at the arrows, you find a matrix scaffold interposed between the muscularis propria and the creeping fat, and we generate a hypothesis that interposed between the two may have a role in creeping formation. And unfortunately, it seems like these videos in this presentation platform are not working. But this is an experiment where we used primary human mesenteric fat that you see on the top left and planted onto a plastic dish, and there was hardly any cell migration out of the mesenteric fat. But when we generated two-dimensional primary human intestinal muscularis propria muscle cell derived matrix, so we plant muscle cells into a dish, we have them produce matrix for 2 weeks, we remove the muscle cell, the matrix remains, and then we put the mesenteric fat on top of it. There was a robust outgrowth of spindle-shaped cells. We characterized these cells. These are adipose mesenchymal stem cells that express PREF1, precursors that migrated out of mesenteric fat in increased amounts in the presence of muscle cell-derived matrix. We then characterized the muscle cell-derived matrix using proteomics, identified fibronectin and decorin as the top two regulated ECM molecules. And decorin was downregulated, fibronectin was upregulated. And fibronectin was found in robust amounts in this matrix scaffold interposed between a creeping fat and the muscularis propria. We then identified how do these adipose mesenchymal stem cells interact with the extracellular matrix derived from muscle cells. We identified which integrins and signaling pathways mediated, and ultimately went to a three-dimensional ex vivo model where we decellularized the human colon, created slices of the matrix underlying the mesenteric fat. The two examples on the right. Unfortunately, also, this is a video that it seems like it is not working, but we then fluorescently labeled pre-adipocytes in red, planted them onto the three-dimensional extracellular matrix, and they move around happily, which is the control. When you then block certain integrin pathways, they stop moving, which you see on the right. The ultimate model is on the bottom left, where we use the three-dimensional decellularized gut slices and add the mesenteric fat directly on top of it. And then we see the adipose mesenchymal stem cells migrating out of the mesenteric fat onto the three-dimensional extracellular matrix. But ultimately, in intestinal fibrosis, as in intestinal inflammation, I strongly believe this is
I think we seem to have lost him for a second. Let's see if we can get him back. We'll be back shortly. Great, Florian. I think you're back now. So
Okay.
apologies for the interruption. All.
No problem. Can you hear me?
Yeah, we can hear you clearly now.
Very good. And where did I stop? Like, where was the interconnection
I
interrupted?
believe it was at slide 48, 49.
This one?
Yeah.
Okay. I apologize. I just kept on talking because for me the template was it all stayed stable. So
No problem. No problem.
All right, let's restart here. So the and thank you all for your patience. In intestinal fibrosis, we strongly believe in a cell-cell interaction model. So immune cell, immune non-immune, or non-immune cell interactions. And I think this all happens in the context of the extracellular matrix and an altered extracellular matrix. So I think we need to see how cell functions in the actual environment. But to come closer to the cell heterogeneity, we generated the first single-cell RNA sequencing atlas of stricturing disease using non-involved, involved non-stricture, and strictured segments from the same patients, and identified tremendous heterogeneity in cell clusters. It's a fairly large atlas, more than 400,000 cells that have been sequenced. I'm just going to highlight the major findings of this atlas, which interestingly I mentioned to you earlier. There's a dramatic thickening of the muscularis propria, but at least in resection tissues, most of the differences in cell clusters and transcriptional profiles in Crohn's disease strictures occurred in the mucosa and the submucosa, but not in the muscularis propria. The differences in the muscularis propria were present, but to a much lesser degree than the mucosa and the submucosa. And I think one major findings of this work emerge as the major signaling cell in stricturing disease. And this is a bioinformatic cell-cell interaction analysis, where on top you see the cell types receiving signal. The top line is health, then CD non-involved, CD involved, inflamed non-strictured, and then on the bottom CDS for CD Crohn's disease strictures. The more the color turns towards red, the stronger is the receiving signal on top. And in terms of which cell types receive signals, it's quite heterogeneous, and they come from essentially all cellular compartments. But when you assess which cell types are sending signals and how this changes and progresses from non-involved towards stricturing disease, these are all fibroblast or stromal cell populations, which is depicted on the bottom. So in other words, the major signaling sending cell type that emerges in stricturing disease are stromal cells. And we then identified selective stromal cells that are either upregulated in number or upregulated in transcriptional activity in the strictured segment, and assessed which genes they share, and among the genes they share, which one are expressed on the cell surface that may be targetable. And what emerged from this analysis was cadherin-11 as a shared cell surface marker, which we then validated with human tissues, and then using primary human cells in vitro with RNA sequencing, proteomics with cadherin-11 activation, cadherin-11 knockdown, and cadherin-11 antibody blockade. And in fact, in vitro, cadherin-11 was anti-fibrotic in primary human intestinal fibroblasts, and the expression in the tissue was highly selective for fibroblasts, which for Crohn's disease, it would be a very new approach, because fibroblasts have not been targeted, and most existing therapies target immune cells or immune-derived factors. We need available antifibrotics in other organs and no antifibrotics in inflammatory bowel diseases. And this is the landscape of antifibrotics across organs, liver, heart, skin, or lung, with the two compounds approved for idiopathic pulmonary fibrosis, nintedanib and pirfenidone, and more to come. So a rich pipeline, but unfortunately, in a stricturing disease, there's no selective antifibrotic or anti-stricture therapy available. And there was a large heterogeneity in definitions and approaches, so we harmonized this by creating a global stakeholder group that participated in expert consensus to standardize definitions, diagnosis, and treatment targets for antifibrotic stricture therapies. And this work culminated in a proof of concept study design how a phase 2 could look like when targeting stricturing disease. And based on this design, we performed a needs assessment, which tools are necessary to run a clinical trial like this. And one of them being a patient-reported outcome tool directed only for patients with a stricturing disease. We went through the FDA pathway of concept elicitation, item generation, cognitive debriefing. This has now been reviewed by the FDA 4 times. We have adjusted the instrument. This is ready for validation in clinical trials, but this is not the only part of the STAR development program, which is now fairly large. We developed an MR enterography stricture index, CT enterography stricture index. We are well advanced in a stricture histopathology index development, intestinal ultrasound index development. We have now a large biomarker development program in which we collaborate with Nordic Bioscience, for instance. And then we have a large program that just started to define the next generation clinical trial population using artificial intelligence, natural language processing, and large data sets in medical centers. So to go back to the map that we sketched, filling up rapidly with preclinical development tools, a validation machine for novel targets, and then the clinical trial endpoint development and validation. There are plenty of examples now for pharma partners working with us for first-in-human studies, and in fact now on multiple occasions developed trial programs with multiple phase 2 studies starting. And one of them, in fact, is starting now for the first trial with Agumab. It has an anti-TGF-beta approach by blocking ALK5 locally in the intestine in stricturing disease. And to give an example how this proposed trial design that I showed you earlier found entry into clinical trials is an example for a trial that Boehringer Ingelheim started with targeting blocking interleukin 36 receptor. This is a pro-fibrotic cytokine. The study started in 100 centers globally, but was then stopped before the first patient was dosed due to portfolio reasons, but helped tremendously in understanding the regulatory landscape and what it would take to get a drug approved in a completely nascent area. So in summary, intestinal remodeling is relevant for Crohn's and ulcerative colitis. It's a dynamic and multifactorial process. The microbiota and inflammation-independent mechanisms contribute to stricture formation. Creeping fat is a novel and exciting tissue compartment to develop novel therapy targets. And then at the end, we talked about clinical trial endpoints, allowing testing of antifibrotics and IBD. This is our group on the left, the Cleveland Clinic Fibrosis group. In the middle, the Cleveland Clinic collaborators. On the right, national, international collaborators. And I particularly point out the STAR Consortium co-leads, J.G. Fletcher and Brian Fagan, we developed this pathway with together. With this, I thank you very much for your attention. Looking forward to Joachim's talk and then the discussion later.
Great. Thank you so much, Florian. All right. I just want to mention that please feel free to use the private chat to put your questions there, and we will use those questions to use them at the end of the webinar session in the Q&A session. Great. Then I would like to invite Joachim to the stage. Let's see. There he is. Great. And Joachim, if you could go into the presentation folder and then open up your presentation from slide 63. It's on the bottom. It says start presentation. Perfect. Oh, we cannot hear you.
Can you hear me now?
Yes, now we can.
Perfect. Thanks. So last presentation. So for those of you who are still here, so thanks for staying. And as said in the beginning, I will be talking about fibro-inflammation, what it is in IBD, and also give some examples of how we can actually quantify it. So I'll start by actually acknowledging a lot of people at Remodeling Bioscience and also many of our collaborators. And also why are collagens important for IBD? And she already talked a bit about this, but just to give some examples again, then for instance, the epithelial cells, they also express collagens, and actually there's 28 different collagens known to man. But also basement membrane collagens like collagen type 4 is important, related also to the epithelium. And then also in the deeper layers of the mucosal wall, we also have collagens like type 1, type 3, type 5, type 11, and type 16, and even also collagen type 6. So there's many different collagens, and they're expressed throughout the different layers of the mucosal wall. And just to give some examples also how these collagens is related to IBD, then since we have the basement membrane, then some of these collagens could be related to epithelial barrier functions, mucosal damage. A variety of different collagens is related here, but of course also in intestinal fibrosis. And also, as Florian mentioned, most of the IBD patients, if not all, they have some kind of some degree of intestinal fibrosis. Important here is also to note that it has also been proven that the fibrosis in IBD have also to some extent been related to non-response, especially to anti-inflammatory drugs. So what is inflammation and fibrosis? So again, Anna and Florian have already talked upon this, but just again to talk more about it. Then of course the inflammation is where we have the immune cell come into the tissue, and they start to simply degrade it and damage the mucosa. And then on the other hand, we also have the myofibroblast and fibroblast that then start to repair it, the damaged tissue, and also if this goes berserk, then it can actually develop into an stricture. And this is a simplified graph of figure of this. So again, these immune cells, they degrade the tissue, but and in this process they release smaller fragments that is then related to this process. And on the other hand, again, the myofibroblasts, they also release pro-fibrotic fragments that can also be reflective of this. And by this, we can actually monitor and also quantify this balance between the destruction of the tissue. So then we ask the question, what is fibro-inflammation? And if we just search for this on the PubMed, then you can see from 2008 until now, there's only a few publications where fibro-inflammation is mentioned. You can see last year that there was 10 publications out there. So this is, and you can say, I think many of us, we agree that of course inflammation and fibrosis, these are intertwined, but it haven't been talked so much about, I would say. And if we then also ask some of our new friend, ChatGPT, then it states that fibroinflammation refers to a condition characterized by a combination of fibrosis and inflammation occurring simultaneously. And then I think that this is summarizing pretty good what it is. And also recently we had a publication also on a review where we talked about or tried to conceptualize the fibroinflammatory axis in Crohn's. Also related that to how it's related to disease activity. So if you jump right into the tissue, then, and again, as also Florian mentioned, then it must start fibro-inflammation and also the fibrosis must start with the inflammation. So we initially see that immune cells, they are being recruited and activated by the pro-inflammatory cytokines, and this in turn leads to an release of proteases like MMPs, but also cellular states that then degrade the tissue, but also calprotectin. And then these fragments are then released, and this can then in turn also trigger a response, a healing or fibrotic response, where we have TGF-beta being released, and this then triggers the myofibroblasts to secrete more collagen, and then also fragments resembling inflammation can also be released, and finally the new collagen will also be cross-linked, and if this becomes too excessive, then the tissue, the fibrotic tissue, can then become really stiff. And then upon this ECM remodeling, these fragments resembling the different aspects, so inflammation will also be secreted into the circulation here. CP9 as an example, but also collagen fragment C3M as a mucosal damage fragment, and finally also ProC3 fragment as a reflective of the fibrogenesis, and finally CTX3 as a marker of resolution of fibrosis. So these, and therefore we can quantify these different processes. And this is also a simplified version of the previous graph or figure, because the epitope matters, and it also says that, or it tells us that these epitopes, they're related to the different immune and fibroblast cells. This simply is reflective of this this ongoing active process. And again, if we limit ourselves to only quantifying the protein, then we believe that we will lose a lot of information. So therefore, it's important to focus on the epitope, as you can see here. By targeting different epitopes, we can actually get a lot of information, so we can quantify the formation of new tissue, degradation, and even also cross-linked by looking at the same one protein, but the different epitopes. And as an example of how we can quantify this balance between degradation and formation, then we can see here with the Crohn's disease patients as an example, where we'll group them according to the CD score. And what we see here is that in the blue dashed line, C3M, so the degradation biomarker, is going up in relation to the severity of the disease, whereas with the formation biomarker per ProC3, we see the opposite. So in remission, patients have higher ProC3, which could indicate more healing, or even in the initiation of the fibrogenesis. So by this, we can actually monitor this balance of formation and degradation. And now for the fibro-inflammatory part, and what we believe here is that this fibro-inflammatory axis at least have these four different aspects. So we have an inflammatory component, and then a mucosal damage component, fibrogenesis and healing component, and finally a fibrosis resolution component. So if we start with the inflammation, here of course we have all the cytokines, chemokines, and recruitment of the immune cells. And an example of how we can quantify this is again the CPA9-HNE biomarker. And again, to talk a bit about it again, calprotectin is of course known to be expressed by the neutrophils, and so is human neutrophil cellular states. And upon the cellular activation of the cells, then it can undergo this activation process called netosis, where it releases all of its cytosolic matter, including the calprotectin, but also the elastase. You can see the elastase in red dots and the calprotectin in the green crosses. And then the elastase start to degrade the tissue, but also calprotectin. And then it releases these, or generates these CPA9-HNE fragments that is released into the circulation. And also, as Anna showed, we have seen that this marker is highly associated with the neutrophil activity, and we demonstrated this in vitro, where we activated human neutrophils. And what we saw in the supernatant was that the CPA9 SNE fragment was only being released by the deactivated cells. And comparing this to a conventional way of quantifying calprotectin, then there was basically no difference between the inactive cells and the activated cells. So this again confirms that we also need to consider these epitopes, because then we also get more information. So again, this confirms that CP9 is a marker of neutrophil activity. We also see that CP9 is related to the activity of IBD. So here's an example of UC, where we can see the full Mayo score, and again we can see a gradual increase in the biomarker levels and correlating with the severity of the disease. Then we also asked the question, because for clinical trials you often want to recruit moderate and severe patients. So we asked the questions whether the CPA9 HNE marker could actually identify this group of patients. So this is what you see on the right side for Crohn's and colitis, and you can actually see that CPA9 was actually fairly good at identifying at least some of the patients in moderate and severe, and could actually discriminate between moderate and severe and remission and mild for both UC and CD. And also we have seen in this marker, and this was presented last year at the DDW with the ozanimod treatment in the introduction phase, where you can see that CP9 was actually more reduced in the ozanimod arms compared to the placebo. And in the bottom, you can also see to the left that the patients who achieved a remission also had substantial more suppression of the CPA9 compared to patients that did not achieve remission. And we also confirmed this in a CD cohort, where these were treated with anti-TNF alpha. We can also see again, patients who achieved clinical remission also had the most substantial decrease in the biomarker. And in this cohort to the right, fecal calprotectin and CRP did not have the same discriminative power to identify or discriminate between responders and non-responders. But at least this also shows, and I think confirms, that CP9 is related to the disease activity potential, also the neutrophil activity, and may be used as a marker to monitor the drug response. After the inflammation in terms of the fibro-inflammation, then comes the mucosal damage. And as an example, we have collagen type 4, and what is also interesting here is that the collagen type 4 actually have 6 different alpha chains, and you can see it here how these are expressed throughout the mucosa here. And I've highlighted the C picture, which is the alpha 3, and it's interesting because it seems that it's only being expressed by the you can say the outer layer of the epithelium facing the lumen, whereas the alpha 1 seems to be more expressed throughout the tissue. So a marker that actually reflects the alpha 3 chain degradation of collagen type 4 we see here on the left. And what is interesting to see, it seems to be more elevated in the mild group of patients here, indicating perhaps this is a measure of the initiation of the flare, or even initiation of the mucosal damage. And this is in a contrast to C4M, which actually measures the collagen type 4 alpha 1 chain. And here this marker was higher or elevated in patients with the severe disease. So we have this difference in the expression of these two markers. And again, it again confirms that the epitope matters. And combined, these two markers, then we actually have a better chance of separating these patients. And also other markers that is related to the mucosal damage. We have seen with C1m degradation of collagen type 1 and the C40 as a T cell activity marker, that patients with the highest levels of these two markers was also in risk of having a fistulizing disease progression. And also we have seen that markers reflecting degradation of collagen type 4 and collagen type 3. We saw that these markers was elevated in patients who did not achieve endoscopic remission, whereas those that did had a lower levels already at baseline. So it seems like we have this two potentially different profiles that could indicate that patients who do not achieve endoscopic remission perhaps have a more severe disease and could again talk into this fibro-inflammatory compartment where they have more inflammation and fibrosis going on. And also we have seen that the markers, other markers is related to the monitoring of the potential response here. So ECM, which is a macrophage activity marker, and also the turnover of collagen type 3 was shown to be also more substantial suppressed in patients responding to Vedo. And also we see this in other cohorts also treated with the Vedo, where again these degradation biomarkers, patients who have lower levels at baseline, and you can see this in the tertiles, that tertile 1, 90%, between 82 to 90% of patients are the ones that benefit the treatment. So again, this seems that the low levels of these degradation markers is beneficial for the patients. And after the mucosal damage, then perhaps the fibrogenesis and healing aspect starts. And again, to talk about how the collagen and the fibrosis affect the response here, then you can see that stagings of collagen type 3 here in infliximab naive and infliximab failure patients, that the failure patients have simply more collagen type 3 stained in all of the different layers here. So again, this talks into that fibrosis must be related to some kind of a non-response for IBD. And therefore, potentially that this could be looking like this, that when we have a response profile, may have more immune cell profile with a more functional epithelium and less severe fibrosis going on. Whereas for the non-response profile, of course, to anti-inflammatory drugs, then perhaps we will see a more fibroblast activity profile with the more tissue stiffness, but also more severe fibrogenesis going on. And to also test some of these markers and relate them to the activity of the myofibroblast, then we also use this Scarnet-Jar in vitro system, which is also a simple system. We use colonic fibroblast, and then we stimulate them with, for instance, TGF-beta and also TNF-alpha. And what we see here is that when we stimulate the cells with the TGF-beta, we can see that for all these markers, the formation of collagen type 1 and 3 and 6 is simply elevated, and it was more substantial with the formation of collagen type 3 quantified with the PRO-C3 biomarker. And on day 4, we then added an ALK5 inhibitor to see whether we could suppress the biomarkers. And you can see here with the green and the dark green lines, we were actually able to completely shut down the production of these biomarkers here. So again, this confirms that these markers, PRO-C1, PRO-C3, and PRO-C6, these seems to be related to the fibrogenesis, and therefore they reflect the activity of a myofibroblast. And this is also confirmed in serous rest setting in these wells, that you can see that TGF-beta treated cells, they have more collagens, whereas the ALK5 treatment simply shuts down this, and you can almost not see any collagens being deposited. And then what is really interesting as well in relation to the fibro-inflammation, then we have some examples here from the dermal space, and here we combined TGF-beta with the TNF-alpha here. So what is interesting, if you look at the black line, this is a combination of TNF-alpha and TGF-beta from the beginning of the experiment, And you can see that type 3 is actually really high, induced by this combination, and it's higher even than TGF-beta alone. So this again talks into this fibro-inflammation being important, and also for the collagen type 6, the combination as such didn't. It was the crossover of TNF alpha to TNF beta that actually increased the formation of collagen type 6. And finally, we also saw the same with the fibronectin, that the combinations of TNF alpha and TNF beta were higher, had increased this release with more potency. So again, I think this confirms pretty good that inflammation and also fibrosis, these are intertwined, and we of course need to focus on this. Examples from the clinic, where we also have seen some markers that is related to the fibrogenesis. ProC11 quantifies the formation of collagen type 11, and you can see here that ProC11 was also elevated in the patients with the stenosis, and also ProC16 was also shown to be related to this, and we also confirmed this marker to also be a marker that can be used for the chronic DSS model. And this is some data actually from Florian's group. What I found interesting here was that they showed that, for instance, if you look at the bottom left, collagen type 6 alpha 3 chain was higher expressed in the IBD, and also what they did was to look into these tissues from IBD, and then the decellularized, and then they added on top of this T cells. And what they saw here, and you can see it on the right side with the white dots, these are T cells attaching to the tissue. So it seemed like that the tissue from IBD with a potential more fibrotic component increased the adhesiveness of the T cells, which could imply that again this fibrosis is related to the potential chronic inflammation that we see in IBD. And the ProC6 biomarker is actually quantifying the collagen type 6 alpha 3 chain. And it's also interesting to see this marker to be also elevated in the patients with stenosis, see on the left, but also we saw that it seemed also to have some prognostic effect, that patients who developed stenosis also had elevated levels prior to this. Finally, we have for the fibro-inflammatory axis, we have the fibrosis resolution. This is where you can see to the right with the green, where we simply remove the old and stiff fibrotic tissue. And therefore, the CTX3 biomarker could be a good choice for this, because this actually quantifies the cross-linked of collagen type 3, and therefore it's the total opposite of the ProC3 biomarker. And also in the Scarlet jar fibroblast model, we observed again by treating the cells with TGF-beta, we induced a higher collagen type 3 from measuring here, as you can see on the left. But when we then measured the cross-linking biomarker CTX3, we couldn't measure anything. Only when we added MMP9 and MMP13, we were able to generate this cross-linked fragment. So this again, I think confirms that at least CTX3 is quantifying the cross-linked collagen type 3. And then to sum it up a bit, then the fibro-inflammatory axis here, we have a inflammatory burden, but also a fibrogenesis burden. So if we look at the inflammation, that might be more in the inflammatory burden compartment rather than fibrogenesis. But of course, this can also lead to this will also lead to more tissue degradation, where some of it is perhaps more related to the inflammation, but some of this mucosal damage can also have high fibrogenesis. And also for the fibrogenesis can also have this high tissue degradation, and is related to the inflammatory burden. But also some patients may also experience that they have perhaps less inflammation still. And finally, again, the resolution here, where maybe we will have less inflammation and also less fibrogenesis, that then allows for the clearance of the old and stiff tissue. And for my last slide here, I just want to finish with this one, because I think it's really interesting with some of these want to say new found cytokines, at least the TL1A seems actually to affect many different cells. And in this example, the TL1A cytokine actually promote inflammation, but also promote fibrosis. So again, I think this again confirms this talk about fibro-inflammation. So I thank you all for your attendance, and looking forward to your questions. Thanks.
Thank you so much, Joachim. Great. Then I would like to invite all speakers to the stage, because we have quite some questions lined up. And for the audience, if you have any more questions, feel free to address them in the private chat, and then we will take those and ask them to the presenters as well. Great. So first question, Florian, what do you believe is the most immediate medical need for the IBD field?
IBD field in general, you mean?
Yes.
I mean, I think it's a very broad question. So probably still the largest unmet need is in having effective anti-inflammatory therapy, simply based on the patient number and the frequency of primary non-responders and secondary loss of responders. So I still think there is a big need, but we are getting better and better in treating inflammation, and that puts on the forefront other complications that are associated with impaired remodeling. So as an example, obviously stricturing disease, perianal disease, so penetrating disease. I do think but very hard to, it's not defined, it's very hard to treat, but still a clinical problem. And then we have other niches, prevention of postoperative recurrence, pouch, acute severe ulcerative colitis. But I think in general, if you ask for the top three, I would say luminal disease, perianal disease, and stricturing disease.
Great, thank you. Joachim, could we replace fecal calprotectin with serum CPA9-HNE, or do you see it as an add-on?
Yeah, so of course fecal calprotectin is have shown to also be a good biomarker, and is of course more specific to IBD. CPA9 is a more systemic marker, but I think the design of it is potentially more related to the activity of the neutrophil. So perhaps in the near future, I don't see that it will be will replace the fecal version, but at least it will complement it, and you can also get more information about perhaps the activity of the cells, because perhaps some of the fecal calprotectin will not be released in patients who have not have the barrier or have the barrier functions, but then perhaps the CPA9 will be released before this. But we will of course see what the future will say.
Great. And a short follow-up question: can the marker also, the CPA9-HNE marker, also be used in rodent models? So is it a translational marker?
Not this specific sequence. There's a mismatch in this sequence to a problem. So not right now, but we are actually trying to also develop a more specific marker for the preclinical models. Yeah.
We will know soon, hopefully.
Yeah.
A question for Florian. Do you believe in one key antifibrotic target, or do we need to target multiple pathways simultaneously, such as matrix stiffness and creeping fat? Or should we even think about combination with anti-inflammatory drugs?
Yeah, so the initial, it's a good question. So the initial approach that we propose, which is how the first trials with anti-stricture therapies are designed, it's a combination of an anti-inflammatory with an antifibrotic. Do we need combinations within antifibrotics? I don't know, but I would assume so, analogous to luminal disease, where I think to expect that one single drug takes care of everything, it's probably expecting too much. So we may end up with combination anti-inflammatories, which is already happening, and combination antifibrotics, but this is not how obviously it's going to start. And then what are the most promising targets? And then I think the most promising still remains anything related to the TGF-beta pathway, because it's so prominent in any investigation across any fibrotic disease. Problem are the side effects, so heart valve issues, skin cancer, systemic autoimmunity, and the way around it could be what is now happening with this Agomab trial. There's public information about with this Agomab trial, where you have local delivery, local degradation formulation with low systemic exposure, and that's the big benefit, I think, of the gut.
Thank you. Question for Joachim. So what biomarkers, so you've shown a lot of biomarkers today, what biomarkers are more specific towards IBD fibrostenosis?
That's a good question. So far we have seen that PRO-C16, that was actually the first one that we saw to be related to stenosis. And it was, for instance, we also saw, compared that in liver disease, and it seemed that in liver PRO-C3 it seemed to be better there. So PRO-C16 seems to be a really good marker for stenosis, and also PRO-C11 and PRO-C6. But I will also say that PRO-C3 also is related to this, as I showed you, the colonic fibroblast also secrete and is really high amount of this PRO-C3. So perhaps PRO-C3 is more related to the fibrogenesis, but not so much the stenosis potentially.
Thank you. Another question for Florian. Could the different subtypes of fibroblasts be the reason for failure of some of the most promising biologics?
So I do think, so what is clear across biologics is that the longer the disease duration, the less they respond. That's the patient response. Why this is, we don't really know. What the data that Joachim presented from our group, we published it this year or last year. We started exploring different mechanisms, and one proposed mechanism, for instance, is that the matrix becomes more sticky to immune cells as disease progresses, and this was the work that Joachim was referring to. And we think, for instance, collagen 6 in this instance has a role, which is in general a very interesting ECM molecule because of its bioactivity. So it has a tail end that is called endotropin. If it gets cleaved off, the matrix molecule becomes highly bioactive in a variety of settings. So this is just one out of many potential mechanisms. I think the matrix could play a role in that scenario. The fibroblasts could play a role in that scenario. I don't know if it's a specific fibroblast subtype, but what struck me in this bioinformatic analysis and unbiased analysis was really that the fibroblasts emerged as a major signal giver in stricturing disease. So that what that suggests to me is it's worth exploring to target fibroblasts selectively because they emerge in tissue damage as the major problem. How that will happen remains to be determined. Cartin 11, obviously, we think is a really nice molecule, but it's not easy to target in vivo. That's no secret. It has been explored in rheumatoid arthritis, and just like with integrins, from a biochemical perspective, not easy to work with. Integrins are targets that could bubble up on top of the list because they're integrins that are stromal enriched or not entirely selective, but are highly upregulated in stromal populations. I think this will be potential avenues, but too early to say. I mean, we just described these fibroblast populations now, and all the rest, a lot of more work needs to be done.
Thank you. There was another question on Vimentin. So in 2017, the University of Alabama identified Vimentin as an important target for controlling fibroblasts, and also showed that it could fully block fibrogenic disease like lung. And then there was another paper that targeted Vimentin, which reduced fibrosis and markers in lung and liver. Have you stained any GI tissues for Vimentin or looked at this correlation in GI tissue?
And did you just raise your hand? Or there was unrelated?
Unrelated.
Yeah, we have done Vimentin stains, and I would be curious to ask you this question because I just recently had a discussion at a fibrosis conference about it, and maybe the same person. We've stained tissues with Vimentin plenty of times in different phenotypes of the gut, and Vimentin is a very widely expressed cytoskeletal component in stromal cells. So when you have an accumulation of stromal cells like in Strickchen disease, we find more expression of Vimentin. We haven't functionally evaluated this. I think it is quite interesting. What would need to be figured out is how would that affect the inflammation or fibrosis broadly, because it's such a ubiquitous component that I think would need to be explored carefully. But then at the same time, with cytoskeletal proteins, and we just presented at ECHO our work on septins and intestinal fibrosis. Not only the amount matters, but arrangement matters, and they have critical functions in vesicle trafficking, intracellular vesicle trafficking, collagen processing, and other things. And so I think in general, as a field, very worth exploring, and in the gut, uninhabited.
Great, thank you. They're saying they're not the same person, but they'll be happy to. We'll make sure to connect with you.
Yes, sure. I'm happy to. But maybe more people are interested in it.
Exactly. Let's see. Just going through the list of the questions. Joachim, do you see different treatments affect different biomarkers in IBD, or is it the same biomarkers that are modulated with all the different treatments?
That's also a great question. So we have seen, for instance, that there's differences in, for instance, C3M and C4M between, for instance, the JAK inhibitors and then also TNF-alpha and the V-beta. So there is definitely mode of actions, as far as I can see, that can affect these different biomarkers here. So yeah, we do see it, and of course, most of these treatments, anti-inflammatory treatments, they of course affect the immune cells. So I will then believe that CP9 would still be a good choice here, because that is by design. It should be also suppressed when you have an efficacious and anti-inflammatory treatment.
Great, and another follow-up question. What technique do you use for decellularization of the series red staining without damaging the structure of the collagens?
That's a good question. I didn't do the work, so I don't actually know. But Florian did the same, or at least someone in his group did. So Florian, do you have any good comment here?
Yes. So it's the techniques are separated by the type of detergent you use, essentially. For instance, sodium desoxycholate is one, which was the one that was most effective for us. And then there's a lot of, so it's detergent washing, and then you remove nucleic acid with like DNases, for instance. And there are a lot of protocols published. Ours are currently in revision, so we hope that they will be available soon. And then obviously happy to share any detail, but we reported in much detail in the paper. But the basis is there's a main detergent, and it takes several days. then the cells go away.
Thank you. And then some more general questions for anybody to address. For clinical trials of antifibrotic trial in IBD, which patients or at which stage of fibrosis should we be targeting?
Yeah, so the FDA has commented on starting with severe populations, so acute patients that have symptoms of obstruction with an imaging confirmed existing stricture, for the reason that everyone wants to do preventive, and that makes a lot of sense, but we don't have a defined population yet that we know. And for that reason, because we all want to target this population, we started this large, what I introduced in my presentation at the very end briefly, very large data AI EMR prediction modeling that includes central reading of images and so on, to get closer to finding this population. Because ideally you want to prevent the disaster from happening rather than having the disaster occur and then try to get out of it. But what we start with early on is with this very progressed populations, and to see, not unlike when biologics were developed, anti-TNF, you start with very sick patients first.
Great. I think we'll end the webinar here. Thank you so much for attending to the speakers and of course for the attendees. A recording of the webinar will be sent to you in a couple of days. For all questions we were not able to address, we'll make sure to address that in the next few days as well by email. And if you have any further questions that come up in the next few days, please don't hesitate to reach out to us. There will be a small survey after this. I end the webinar, so please fill that out if you have the chance to. Thank you so much, and have a wonderful day.
Thank you all. Nice meeting you.
Bye. Bye.